Respected faculty members, students, and my dear colleagues, good morning to all of you. Today I am honored to present our CRT funded project titled Bioactivity Guided Discovery and Characterization of Novel Antimicrobial Metabolites from Syzygium praecox. I am Mohammad Jain Hussain, lecturer of Department of Pharmacy and serving as the principal investigator of this project. My co-principal investigators are Dr. Chaudhary Faiz Hussain, Dr. Muhammad Muhyiddin Kobir, sir from GEB, and lecturer Muhammad Johirul Islam from Department of Pharmacy. Before any phytochemical or biological investigation, correct taxonomic classification of the plant material is absolutely essential. Here is the taxonomical classification of Syzygium praecox. It belongs to family Myrtaceae, the same family as of clove, and it belongs to the genus Syzygium, which is one of the largest genera of flowering plant with over 1200 species worldwide. And geographically, this species is found across a broad region from East Nepal through South Central or Southeast China and into Indochina. Now we know our target plant. Let me set the scientific context for why this research is necessary. Let me begin with the situation in developing countries, particularly Bangladesh. Countries like Bangladesh face the highest burden of antimicrobial resistance due to misuse of antibiotics, easy availability without prescription, and substandard formulation. Now, let me share some alarming global statistics. AMR is recognized by the WHO as the top global health threat with projections of 10 million deaths annually by 2050 and 4.95 million deaths reported in 2090. Now, let me bring the focus to our plant Syzygium praecox. This plant is traditionally used for diarrhea, dysentery, allergy, inflammation, and skin infection. However, no bioassay studies exist to scientifically validate these claims. Moreover, more Syzygium species demonstrate diverse antibacterial activities. Medicinal plants offer lead compounds for developing novel antibiotics with fewer side effects, supporting the potential of Syzygium praecox. Now, let me clearly articulate the problem of our research aims to solve. First, no scientific validation. As I mentioned earlier, Syzygium praecox has been used in traditional medicine for diarrhea, dysentery, skin infection. However, no bioassay studies exist to scientifically validate the antibacterial properties of Syzygium praecox. Second, a completely unexplored species. To date, no published antibacterial studies have been conducted on Syzygium praecox anywhere, creating a scientific knowledge gap. Third, unknown chemical profile. The specific secondary metabolites responsible for any potential antibacterial activity remain entirely unidentified and uncharacterized. Fourth, missed therapeutic opportunity. Without systematic investigation, a potentially valuable source of novel antibiotic compound from Bangladesh's biodiversity remain untapped. So let me summarize the problem in one sentence. We have a plant with traditional antibacterial claims, but no scientific validation, no published studies, an unknown chemical profile, and a missed opportunity for drug discovery. This is the gap, and this is what our research will fill. Our project is designed to deliver multiple outcomes. First, discovery of novel bioactive compounds. We expect to identify previously undocumented phytochemicals from Syzygium praecox with high potency antibacterial potential. We expect to design a comprehensive map of molecular mechanism and bioactivity profiles, providing a scientific clarity on how these compounds combat resistant bacteria. And we expect to create a library of standardized lead compounds optimized for downstream drug development and clinical trials. Fourth, methodological innovation. Beyond the compound themselves, our project will establish optimized protocol for the efficient extraction, isolation, and structural characterization of plant-based metabolites. Next, global health impact. Contribution to UN SDG Goal 3 by fostering sustainable research that ensures long-term health and well-being through new AMR solution. And finally, potential synergistic effect. We expect to identify potential synergistic effect with existing antibiotics, which will help in combating antimicrobial resistance. Our research is guided by an overarching main objective, that is to apply a bioactivity guided isolation strategy to identify and characterize antibacterial compounds from Syzygium praecox leaf, focusing on novel agents active against selected multi-drug resistant pathogens. Let me break this down into 5 specific objectives. First, antibacterial evaluation. We'll evaluate the antibacterial activity of isolated compound against standard gram-positive and gram-negative strains. We'll prepare a crude leaf extract via cold maceration and fractionate it using polarity-based solvent sequence. We'll also isolate constituent only from the most active fractions using bioactivity-guided chromatographic methods. Once we obtain the pure compounds, we'll elucidate the structures of isolated compounds using high-resolution mass spectroscopy and 1D or 2D NMR with advanced analysis reserved for novel compounds. And fifth, for any novel or highly potent isolates, we will determine the minimum inhibitory concentration and minimum bactericidal concentration of any novel or highly potent isolates. This slide presents our literature review. A comprehensive review of the scientific literature reveals that the Syzygium genus is a rich source of bioactive compounds with diverse therapeutic potential. The literature provides strong scientific foundation for our hypothesis. So here we have shown our proposed methodology for our research. First of all, we will collect the plant with proper identification. After drying, cutting and crushing, we will provide this in cold maceration. After methanolic extraction, we will perform solvent partitioning with different types of solvent like anhexane, chloroform, ethyl acetate, dichloromethane, etc. Then we will obtain the fractions in different test tubes, and for each of the fraction we will undergo antimicrobial assay using the minimum inhibitory concentration assay, and the compound which will have the lowest MIC, that is which will have the highest antibacterial potential, will be the most active fraction, and using the most active fraction we will conduct column chromatography, and by conducting the column chromatography we will collect different fractions of the active fractions. We will collect some subfractions of the active fraction, and using the subfraction we will conduct antimicrobial assay once again, and we will perform the bioactivity screening, and after that we will perform the TLC and SPLC isolation of the fractions. Okay, we will isolate the active compounds by TLC or SPLC. Okay, after the isolation we will determine the exact compound by high resolution mass spectrometry and NMR. And by this, we'll ultimately get the potential antimicrobial agents obtained from Syzygium praecox with its structure. So this is the overall methodology for our research proposal. So these are the significance of our research proposal. This research aims to extract and isolate secondary metabolites from the leaves of Syzygium praecox. By identifying the secondary metabolites and analyzing their chemical structure through spectroscopic methods, the study seeks to provide scientific validation of the traditional use of this plant. And additionally, the antibacterial activities of these isolate compounds will be assessed, which may contribute to the discovery of bioactive compounds with potential therapeutic application and ensure the safety of the development of antibiotics. Now, our research is structured around different specific research questions. These questions directly emerge from the gaps we identified in the literature. First, which specific solvent fraction of the Syzygium praecox leaf extract demonstrates the most potent in vitro antibacterial activity against defined panel of standard strain, including Staphylococcus aureus, Bacillus subtilis, E. coli, and Salmonella species? This question is fundamental as it directs our entire isolation strategy. This answer will tell us exactly which fraction to pursue. Second, can bioactivity guided fractionation of the most active lead fraction lead to the isolation of pure compounds? This is our core methodological question. We are asking whether our approach following the activity at every step will successfully yield pure active compounds. Third, what are the chemical structure of the isolated compounds as determined by spectroscopic techniques, and are of these compounds novel chemical entities? This question addresses the novelty and scientific contribution of our work. If we discover novel compound, that would be a significant contribution to natural product chemistry. Fourth, does polarity guided fractionation significantly influence the yield and bioactivity of isolated metabolites? This answer will help optimize future extraction protocol for this and related species. Fifth, what is the in vitro antibacterial efficacy of the isolated pure compounds, particularly any novel compounds against the panel of bacterial strain? MIC and MBC are the standard parameters for comparing potency with existing antibiotics. Sixth, which class of secondary metabolites is primarily responsible for the observed antibacterial activity of the most active fraction? So this will help in identifying the active class, will help in the mechanism and guide future isolation efforts. Seventh, do the isolated compounds show selective activity against gram-positive or gram-negative bacteria? 8. Is there any synergistic antibacterial effect when isolated compounds are combined with standard antibiotics? This is a clinically important question. Even if our compounds are moderate active on their own, they might enhance their activity of existing antibiotics, potentially reviving drugs that has lost their effectiveness due to resistance. So there are some risk assessment during our research project, like degradation of heat level bioactive compounds during extraction and processing. Then there may have some low yield of active metabolites after fractionation and purification. There may have contamination during microbiological assay, which may lead to false antibacterial results. There may have some instrumental limitation or errors in spectroscopic or chromatographic analysis. There may have some difficulties in isolating pure compounds from complex fraction. There may have some variability in antibacterial activity due to experimental condition and limited resource of advanced structural elucidation of novel compounds. So these are the risks. However, to mitigate the risk, we will take these actions, like we will have controlled extraction condition and temperature regulation to preserve compounds. We will perform bioactivity guided fractionation to focus only on active fraction and avoid resource waste. We will apply strict aseptic techniques and standardized protocols during antibacterial testing. We will calibrate and validate analytical instrument before use. We will perform stepwise purification with continuous SPLC or TLC monitoring. We will replicate assays and standardized condition to ensure reproducibility. We will have strategic use of advanced spectroscopy only for potentially novel compounds to optimize resource. So no study is above of limitation. So our study has some limitations, like no human clinical trial would be conducted in this research, and the research doesn't elucidate the actual mechanism of antibacterial activity. However, some future research can be conducted by taking idea and knowledge from our research, and our research can be further broadened into these advanced investigations, like uh, future further investigation to identify molecular mechanism and isolate specific bioactive compounds responsible for the activity. And moreover, isolated bioactive compounds could be developed into pharmaceutical formulation as alternatives treatments to synthetic drugs. In conclusion, this study highlights the potential of Syzygium praecox as a source of bioactive secondary metabolites with antibacterial activity. Through bioactivity guided isolation, purification, and structural characterization, this research aims to identify novel compounds that could contribute to the development of new antibiotics. While further studies are needed to elucidate the precise mechanism and clinical applicability, these findings provide a scientific basis for the traditional medicine use of this plant and open avenues for future drug discovery. So these are the references from where we took necessary informations. Thank you for your kind attention. I welcome your question and feedback. Thank you.